There we go. Yes, almost. Just thinking about it. So we left off Monday talking about ATP. So we'll kind of, since it happened at the end, we'll kind of review it just a smidge, right? Just kind of remind ourselves what we were talking about. Let's see. I think it's bright enough outside that we can all do this. Nope. There we go. That's a little better. Right, so we're talking about ATP, adenosine triphosphate, right? So it's just an adenosine, right? A sugar with an adenine and the 3 phosphate groups, right? And so ATP in your cells is used to power non-spontaneous reactions, right? So endergonic reactions are ones that, right, on that kind of free energy graph, they're those uphill reactions where you need to put energy in to make them go. And so your body uses ATP for that, and it does it, right? We've got these 3 phosphates together with these really unstable bonds. And so if we break one of these bonds, 1, because they're unstable, they break really easily. And then 2, because they're these kind of tense, unstable bonds, they release a lot of energy. So we've got this kind of exergonic, spontaneous reaction in hydrolyzing ATP, right? Using water to break a phosphate off that can then be used to power reactions, metabolic processes within the cell, right? And of course, when that happens, right, we add water, we've got a phosphate that breaks off, we're left with an inorganic phosphate, this adenosine now diphosphate, and then that released energy, yeah? And usually that phosphate that gets broken off gets stuck to, right, some part, right, an intermediate molecule in the reaction that we're powering. So if you remember the sodium pump, we had ATP that powered the pump and made it open. And so when that happened, when we broke that phosphate off, it stuck right to the pump. We kind of saw that in that diagram. Right? So it's just kind of normally how ATP behaves, and it's powering these non-spontaneous reactions in the cell. Right? So this is one way that we're making things go metabolically, right? The other way that we're making reactions go, right, because we can power these non-spontaneous reactions, but we also have a bunch of spontaneous reactions that we want to kind of keep moving a little more quickly. Oh, I keep forgetting that the slide is, I've done this twice today. Right? And this idea that ATP gets reused in the cell, right? Because you use ATP, you break off a phosphate, you've got adenosine diphosphate, and your cell just takes that ADP, pops another phosphate on it, and sends it back out, right? So it's just happening over and over again, right? Your cell synthesizes ATP from ADP and phosphate, sends it out in the cell, it energizes some reaction, comes back, you make it again, right? So it's just ATP is being recycled and cycled over and over and over again, right? And pretty quickly, so that your cell really at any given point in time only has a few seconds of ATP available, right? So it's just kind of happening quickly and repeatedly, basically. And so that is kind of the downfall is ATP. It's basically, right, it's a one-off. Once you pop that phosphate off, you've got to go make it back into ATP again. It can't do anything, can't really do much else at that point. Right. So ATP is cycled in the cell. Now, powering exergonic reactions, right? Exergonic reactions are reactions that will happen spontaneously, but we can make them faster, right? Because they're downhill reactions, but there's always this little bump at the first, right? You've got to kind of give them a little nudge to get them going. And we can reduce that activation energy, right, is what that's called, by using enzymes in biochemical pathways. And so basically, when we start going through all these metabolic pathways, respiration and photosynthesis and all these things that are going on, they're almost entirely powered by enzymes, right? There's going to be enzymes at every single step, kind of moving things along. So we're going to spend a little bit of time before we get into the chapters on respiration and metabolism, talk about enzymes, what they look like, kind of how they function, how they work, how they're controlled, right? And so that's kind of how these last couple sections of chapter 6 round out. So, mentioned when we talked about proteins and when we talked about kind of energetics generally, enzymes function to lower activation energy, right? So you've got a reaction that is spontaneous, right? This kind of energetically downhill reaction, but there's always this little kind of bump at the front, right? And so enzymes basically kind of flatten that out so that reactions will happen more quickly, right? And they do this kind of grabbing onto molecules and stressing these bonds. So they have very unique shapes, right? Every enzyme generally has one very singular specific job, right? It is a specific shape to bond to this specific molecule or molecules and make it do this specific thing, right? So when we talk about enzymes, kind of everything revolves around the shape of the enzyme and things that are affecting the shape of the enzyme. And these enzymes bind to substrates, right? So every enzyme has a specific spot where a molecule will bind to it. And we call that molecule the substrate, right? And so they can bind one substrate and kind of change how that molecule is kind of arranged. They can bring in two substrates and attach them together, right? And so we talked about this, right? Because enzymes will kind of do reactions in both directions, right? They bring things together, they take things apart in that kind of back and forth way that reactions work, right? And so they do this by grabbing these molecules and stressing the bonds so that these reactions will happen more readily. All right. Now, different than when ATP is used to push kind of energize fuel or power reactions, enzymes are not consumed, right? They're more just like a tool, right? Like a wrench or a set of pliers or something, really more like a wrench, right? Because they're kind of like a specific size, right? But they're not consumed or changed, right? They're a specific shape. They do their job to this molecule. They leave that molecule, go to the next one and do it again, right? And so they're not changed in reactions. They are just catalyzing reactions over and over and over again as they encounter their appropriate substrates, right? The molecules that they're meant to do reactions with. So they do this, right? Because they're specific shape, they have a specific spot that is a specific shape for a specific molecule, right? And this spot, this site on the enzyme, right, on this big globular protein, is called the active site, right? It's the site where the thing is going to happen. So you've got this active site, this cleft or pocket that is shaped like some molecule that the enzyme is going to catalyze. And so this is where the substrate binds to the active site. And when you have an enzyme with a substrate attached, it's called an enzyme substrate complex. Sure. Now, you have an enzyme with an active site, something attaches to the active site, you've created this enzyme substrate complex, and then catalysis happens, right? And catalysis, right, catalyzing some reaction, requires that the substrate fit to the enzyme really, really, really well, right? So this substrate, whatever little molecule that this big old enzyme is worried about, right, has to fit very precisely into the active site. Now, we've talked about proteins and how they kind of fold and move. And so there's a bit of kind of plasticity in these big protein molecules. Yeah. They're not rigid, like locked in crystalline molecules, right? They got a bit of wiggle to them, right? And so when this substrate kind of attaches itself in there, it causes a change in shape, right? That a conformation change, right? We use that with these proteins when proteins start changing shape, right? So the substrate binds in there and the protein the enzyme kind of reacts to the fact that the substrate is bound in, and it will frequently kind of wrap around and tighten its binding to the substrate. And we call this induced fit, right? Because the substrate binding induces the enzyme to change shape and fit even more tightly. Yeah. And so sometimes, because the protein is going to move and change shapes, it will actually facilitate, if this is a reaction that needs multiple substrates, right? The enzyme kind of attaching to substrate A kind of opens up the active site for substrate B, right? So you can kind of do these things. It kind of helps things stay in order, in a sense, right? So we have this idea of induced fit where proteins they've got enough plasticity in them to kind of adjust their fit to a given substrate. All right. This is just kind of a diagram from your book that kind of walks through an example that we mentioned when we were talking about carbohydrates, right? We talked about glucose and that glucose can't kind of like float through your body on its own all free. It's got to kind of be chaperoned until it gets to where it's needed, right? So you tend to have sugars moving through your body as sucrose, right? It's a glucose and fructose bonded together. But then when that glucose gets to where you need it, right, you need to break the glucose back off so you can use it for respiration, for metabolism, right, to get the energy out of it. So to move it, right, it floats around as this disaccharide, sucrose. And then wherever it's needed, there's this enzyme sucrase, right? So this substrate's going to bind in, right? This substrate's going to bind into the enzyme, these very specific active sites. Enzyme's going to, right, you kind of see, kind of grabs onto them, right? It's a hydrolysis reaction. So we're going to add water. We're going to stress this bond. These two sugar rings are going to break apart and then be released, right, as free glucose and fructose. Wherever it is that they're needed, they go to be used. And once those molecules are released, the enzyme opens back up and is just exactly as it was when the process started. So it goes to do that reaction again. Right? And so this is generally kind of any enzyme we're talking about. This is just the general process of steps that they're all kind of going through at any given point in time. It's a fairly simple one. Some of them do more complicated things, but basically it's just this kind of a thing, right? This catalytic cycle. So breaking down, this disaccharide into two monosaccharides is an example of a catalytic cycle. Yeah. It's a good question. I honestly have no idea. Yeah, I don't know. I assume not very long. I'm sure I could look it up. I'm sure somebody knows. Yeah. All right. Forms of enzymes. Okay, so we've got these enzymes, proteins. And they're kind of everywhere, right? Because every biochemical pathway, every kind of metabolic little process we've got going on relies on chemical reactions, which generally rely on enzymes. So you kind of basically, if you're looking kind of, if you were traveling through a cell, enzymes are everywhere. They're suspended in the cytoplasm, right? They're embedded in the organelles, right? Things like your endoplasmic reticulum, right, that are building proteins and lipids and carbohydrates and putting things together, right? That's all enzymes doing that, right? And so they're embedded in the organelles, they're floating in the cytoplasm, they're just kind of everywhere. They can also, so they can float around singly, they can also form these multi-enzyme complexes, which are non-covalently bonded assemblies, not that you necessarily need to know that, but that these enzymes will come together. We'll kind of form these bonded groups of enzymes, right? And each of those enzymes is generally catalyzing some step in a process, right? So maybe we've got a series of like 6 chemical reactions that get me from molecule A to like molecule H that I need for something. And so frequently when you have these kind of serial processes, you'll have all these enzymes together. They kind of make a little factory, like a little assembly line, right? So they're all kind of attached together. They can be quite large, right? The dehydrogenase multi-enzyme complex is a really big one. It's actually 16 protein subunits all fitted together. So kind of this giant enzyme factory, kind of running this whole pathway kind of in-house, right? Not that you need to memorize dehydrogenase multi-enzyme complex, it's just an example. Falls into the maybe good for Scrabble category of words. But your cells do this for a few reasons. There's kind of a few benefits to doing things in these multi-enzyme complexes, right? So if you kind of have everything localized, right, where substrate A comes into the first enzyme and becomes molecule B, which is the substrate for the next enzyme, right? You kind of keep everything together and you can make these processes more efficient, quicker. You can increase the rate of these enzyme reactions because everything all the enzymes needs is like all right there in the same place. You can prevent unwanted side reactions, right? Because Another thing to remember, right? Enzymes they're not like little Pac-Mans. They're not out like looking for their substrates. They're just kind of floating around open. And if a substrate bumps into them, then it reacts, right? So if you have kind of all of the enzymes together and all of their associated substrates are then in the area, you can kind of make happen what you need to happen versus having one of those substrates float off, bump into some other enzyme and go down some other pathway that you don't intend. So it just kind of makes things a little more organized. And then you can kind of control these pathways as a unit. We're going to talk a bit about how enzymes get regulated, right? And so again, if they're all together, you can kind of, if you need to turn off a pathway, say, right, you can kind of shut down a pathway as a unit because it's all in this one single complex together. All right. We also have enzymes that are not proteins. Recently, scientists have discovered that there are some enzymes that are actually made of RNA, right? DNA and RNA, right? So you have, they kind of look and work like little enzymes. They do the same thing, but they're just made of a different kind of molecule, right? They're made out of this nucleic acid instead of a protein. And so they do intermolecular catalysis, which is what we were just talking about, right? They catalyze reactions on other molecules. They don't change, right? They do that kind of like, they are a tool in facilitating reactions. But unique to them, they also do intramolecular catalysis, which means they can also do reactions on themselves, right? They can kind of change and conduct reactions like on their own molecular self, which is kind of bananas. And this leads to, if you're kind of interested in like early Earth sort of theories, right? One of them is this kind of like RNA start to life, right? Because with RNA, that can carry all the same information that your DNA does. And turns out it can also catalyze reactions, both on other molecules and on itself. So really RNA is kind of like this, becomes this Swiss Army knife kind of molecule that can replicate itself, fix itself, and catalyze other reactions, which is kind of bananas. The other end of this then means, conclusion next, that ribosomes that we've spent a lot of time talking about and will continue talking about are actually ribozymes. Right? They're actually RNA enzymes. All right. So enzymes are everywhere. They're usually made of protein. They can also be made of RNA. Cool. Now, enzymes are existing and functioning in your cell, and there are a lot of factors that affect the way that enzymes work. So we're going to spend a little time walking through that. So enzyme function, we talked about this when we talked about protein formation, all these things. There's a lot of things that kind of affect how proteins fold and how they behave, right? And so basically those things we listed when we talked about proteins, we're going to list again, but kind of specifically framed in this kind of enzyme catalytic mindset. One is just the concentration of substrates, right? Enzymes aren't searching for substrates, they just bump into them. So if an enzyme catalyzes substrate A and there's a whole lot of A around, it's more likely to bump into the enzyme. The reaction's more likely to happen. So enzyme function will increase if there's more of its substrate around. Fine. We'll walk through the rest of these as well. So temperature next, right? We've talked about temperature and chemical reactions before. Right? You raise the temperature, chemical reactions happen faster, right? Because you're inputting energy. Same thing for enzymes, right? Because enzymes are doing chemical reactions. You add a little more energy, reactions go faster up to a point, right? So you add heat, reactions go faster and faster and faster, but there's a point where it'll level off and actually drop, right? But that point, that highest point is the optimum temperature for a given enzyme. Yeah. Right. Now, if we think about how proteins work, it makes sense, right? Because you've got these long strings of amino acids. They're all kind of folded up and hydrogen and ionically kind of bonded and stuck to themselves to make some sort of shape. And so if they're cooler, right, if the temperature is below the optimum temperature, right? Bonds kind of become more rigid, right? Molecules aren't moving as much, atoms aren't moving as much. And so then your protein becomes like really fixed. You can't have induced fit if the protein is really, really rigid, right? So if it's too cold, the protein gets too stiff, it doesn't fit its substrate very well, it's not going to function very well. If it gets too hot, right? What happens to a protein when it gets hot, like too hot? What? Yeah, melt. What was that? There was a word for proteins getting too hot. Remember that one? That's what's happening when you fry an egg and it turns white. Denaturing. You're denaturing the proteins, right? Because as you heat it up and all of the atoms and molecules get more and more excited, right? That protein gets real loosey-goosey, right? And then you have the opposite problem. The protein is too like splayed out. It may, if it's hot enough, it'll just start falling apart, right? Those proteins start unfolding when they get too hot, right? And so if your enzyme can't maintain its shape, that's like the whole bag with an enzyme is that it's got to be a specific shape. So if it gets too hot, also won't work. So there's kind of this optimum range of temperatures where enzymes function best, which happens to be between 35 and 40 degrees Celsius. What is the internal temperature of your body? Roughly, on average. 97, 98, or? There you go. Between 35 and 40 degrees Celsius, right? So, right, we talked about homeostasis before, this environment that your body, all your cells are maintaining to maintain enzyme function. So your body maintains a temperature where all of your human enzymes function best, right? So any organism's normal temperature is the temperature at which their enzymes are best functioning. They'll always line up, right? If you're a cat, it's going to be like 102 degrees or something. Right, so optimum temperature. Another factor is pH, which makes sense, right? Again, these proteins are folded up. There's all these kind of ionic bonds happening, right? And so if you dump a bunch of protons in there, it's going to screw with all of those bonds that are keeping that protein in a specific shape, right? The protein and its shape and its folding is going to react to the concentration of protons around it. Again, if you mess with the protein shape, with an enzyme shape, it doesn't function very well. So, shocking, the optimum pH for an enzyme is between 6 and 8, which is also the pH of your body, right? Your body metabolically puts a lot of effort into making sure that you maintain an environment where all of your enzymes are working kind of at their best all the time. Ironically, weirdly, circularly, they need the enzymes to do the metabolic processes that keep your body at the temperature that the enzymes want. It's very circular. Exception pepsin, right? Things happening in your stomach, which is incredibly acidic, pH of like 2. So you do have some specialized enzymes that are made to work under extreme conditions. Pepsin is one of them. All right, so last one. So enzymes react a lot to their environment, the amount of substrate, the temperature, the pH, but there are also other things, other molecules floating around in your cells that will affect the way these enzymes are working. Will regulate the way the enzymes are working, as it were. So we've got regulatory molecules of a few different flavors, right? And so these regulatory molecules are able to bind to the enzyme, right? When things bind to an enzyme, it changes shape. If you change an enzyme shape, you change how it's working, right? Two major categories will be inhibitors and activators, right? Things that inhibit enzyme function and things that activate enzyme function. So we'll do inhibitors first. So inhibitors decrease enzyme activity. They inhibit it in a couple ways. You have these inhibitor regulatory molecules, and they can either be competitive or non-competitive, right? And both of these molecules are working to stop the substrate from binding to the enzyme. Right? A competitive inhibitor, right, stops a substrate from binding to an enzyme by fitting into the active site, basically blocking the substrate from binding, right? So they're competitive. They compete with the substrate for the active site and then get in the way, right? Stop the enzyme from doing its job. You can also have non-competitive inhibitors. So these are also inhibiting molecules, but they don't bind to the active site. Instead, they bind somewhere else on the enzyme and just make it change shape so that it doesn't work anymore, right? So they just kind of tack on away from the active site, cause a conformational change, right? Change this enzyme shape so that the substrate no longer fits. So this enzyme no longer works. Now this spot, we'll talk about a bit more, where it binds is called the allosteric site. We'll talk about the allosteric site a bit more, right? So we've got inhibitors that can either compete with the substrate, bind to the active site, or bind somewhere else, change the enzyme shape, make it stop working. Now, these non-competitive inhibitors that bind to allosteric sites can do kind of a couple of things. Like you can kind of think of these allosteric sites as either like on switches or off switches in a way, right? And so we have enzymes that can exist as active or inactive, right? Depending on whether they've been flipped on or off. And those enzymes are called allosteric enzymes because they have an allosteric site. Cool. So you can have allosteric sites that are like this chemical on-off switch. And so if you have an allosteric inhibitor, right, it basically hits, it's an off switch then, right? So an allosteric inhibitor will bind to the allosteric site, turn the enzyme off. Now the enzyme is off, doesn't work. Conversely, right, we've got allosteric inhibitors. You can also have an allosteric activator, right? So that's where on an enzyme, it's got an allosteric site, but in this case, it's an on switch, right? So kind of when it's on its own, the enzyme is kind of by default off, by default not working. We can have an activator that binds to the allosteric site and turns the enzyme on. So in that case, that little molecule is an allosteric activator. It activates the enzyme. Cool. Right? All right. Now, most enzymes kind of function on their own and do their own thing, but there are some enzymes that need kind of assistance to work, right? And so these things, these other molecules and things that kind of help enzymes along, kind of help make them work, are called cofactors. So they're just additional chemical components that attach to the enzyme so that it can work, so that it is functional, right? Couple kinds of cofactors. First one is inorganic cofactors, right? And so they're ions, right? So they're little charged atoms that attach in and kind of draw electrons away from covalent bonds, right? Which is going to disrupt bonds, which is kind of one of the things, right, that enzymes are trying to do. So you can add these ions that will pull electrons away from the covalent bonds, right, and kind of help lower activation energy and cause some reaction to happen. So these are things like zinc, molybdenum, manganese, right? All of these things, all these cofactors are going to sound very much like everything that's like in your morning vitamin. Going to be these cofactors. So zinc, molybdenum, manganese are some examples of inorganic cofactors. You can also have non-protein organic molecules that kind of do this work. Those are just called coenzymes. And so these would be things like B6 and B12. Vitamins B6 and B12 are organic molecules that help enzyme function. Right. So we can have kind of other molecules that help enzymes to function, kind of outside of regulatory molecules. All right, which wraps out 6.4, takes us into the last section of this chapter. So now we're going to kind of go more broadly into metabolism. All right, we've got enzymes, we've got multi-enzyme complexes, we've got ATP, we've got all of these pieces. We're going to talk a bit about metabolism, right, before the last two chapters of this unit, which are on respiration and photosynthesis, which are huge portions of our metabolism, right? So we're going to talk about metabolism, we're going to talk about pathways and kind of how these pathways evolve a little bit, how they kind of come together. So starting with metabolism. Metabolism is just all the chemical reactions that your body is doing, right? So if you have an organism, every chemical reaction that is going on in that organism's body together, right, kind of as like collectively are its metabolism. Yeah. So we've got all these chemical reactions. They can be kind of divided into two main categories. They can be either anabolic or catabolic. Subsequently, we can refer to them as anabolism or catabolism, right? So anabolism are any of the reactions that are using energy to build molecules, right? Because your body stores energy in these bonds, in these molecules. So your anabolic processes are making those bonds, using energy to make those bonds to store energy. Versus catabolism, which are all the reactions that are then harvesting the energy stored in those molecules by breaking them down. All right, so we're building molecules to hold energy. We're breaking molecules to get energy and use that energy to build new molecules, right? Pretty much all of these things are very cyclic. All right, so all of your anabolic reactions and all of your catabolic reactions together are your metabolism. Now, in your cells, in your body, you've got thousands of enzymes going through all of these different pathways, right? Catalyzing loads of things all the time. Many of these reactions actually occur in sequence. It's not like your body just kind of does like one little A plus B is C reaction and it's done with that, right? It's frequently A plus B makes C and I use C and D to make E and then E turns into F, right? It's like, it's a series of reactions, right? And so we call these series of reactions biochemical pathways. Yeah. And so it's just that, right? An enzyme catalyzes something and the product of that reaction is then the substrate of the next reaction, plugs into a new enzyme, creates a new product. That product is the substrate of something else, right? And we kind of move these molecules through different things to make whatever it is that your body is trying to get to, right? Of building or breaking, it depends, right? We'll see both as when we go through respiration and photosynthesis, right? In photosynthesis, basically we're going through a long pathway to build a glucose molecule. When we go into respiration, we're going through a long pathway to break down glucose, right? So we want to store the energy, and then when we need to use the energy, we've got another pathway that's going to break it down and harvest the energy from it, right? And so all of these kind of series of reactions are biochemical pathways. How did we end up so full of so many complicated processes? It seems absurd and ineffective. And in a lot of ways, when we kind of start breaking down things like respiration, it's actually not very efficient. So how would you end up with a process that is not efficient, right? And this is that thing where we have to kind of remember when we're talking about things that are evolving, right? An organism evolving is not like building an airplane, right? I build an airplane and I'm like, I think I can make a better airplane. I don't start like nailing things onto the old airplane to make it better, right? You like chuck it and you make a new, better airplane. Your body can't do that, right? My body can't like chuck itself and build something else. It's not how it works, right? So all it can do is kind of like that dumb way of fixing airplanes, like where this airplane's not really good, so I'm just gonna like nail another board to this piece and hope that helps. And if it crashes, then that airplane goes extinct. I don't know, that's a weird metaphor, but it's what I mean, right? And so how do we get these like complicated things? It's because that's how evolution works. Like you have a thing that's working and something's surviving and suddenly it needs to fix a problem. And I'm like, okay, this kind of fixes the problem. Well, now I've got a new problem. And like that kind of fixes the problem. And eventually you end up with this really cumbersome process, right? And so your body is basically kind of filled with cumbersome processes, right? So example, simple one-step reaction. Maybe we imagine some little single cell prokaryote somewhere in some little puddle, right, doing its thing. And to survive, for whatever reason, it needs this thing H, this substance H. All right, well, to get H, it's got a little enzyme that'll take F and G and turn it into H. Cool, happy little cell doing its thing. Well, what if this cell does really well, kind of proliferates in the environment, and they consume all of F? Well, crap. All right, who's going to survive? If there's no F, we all just die because we need H, unless some organism, by some random mutation, kind of picks up the ability to turn this other thing in the environment, molecule E, into F. Cool, I can catalyze E into F. If I can take E from the environment and make F, I can add that to G and get H. And I don't go extinct. The organism now, organisms that didn't mutate, that can only do this, they die out. Organisms that mutated can now do this, they survive, they carry on. But planet's been around for a while. This happened a lot. Eventually, right, you end up with something much more complicated. We ran out of E, then we ran out of D. All right, now we can make C into D into E into F. Add F to G and get H. Right, and so this is kind of how you get these complicated pathways. Right, so when we start walking through respiration, you're like, why the heck would my body do this way? This is dumb. It barely makes anything. Because you're kind of working with this sort of a system. Kind of inherently, it's a little clunky, for lack of a better word, right? And so this is kind of how these pathways evolve over time. So if you want to see kind of like a, right, so you kind of end up with this sort of thing. You've got some cell, and in its membrane, it's got these series of enzymes, right? It needs this. This is what we're trying to get to. And over time, right, kind of back to front, it has built up this long process, right? This is what we can get from the environment. Through a series of reactions, we can get this thing that we need to survive. Right? You get these complicated pathways. Now, you probably don't want, right, if you're in a factory, you don't just run all of the conveyor belts all the time ad nauseum without any sort of control, right? Occasionally you need to make more of this, occasionally you need to make more of that. So your cells need some way, kind of similarly, they don't need to run everything all the time. They've got to have some kind of way to control, right, kind of like which little manufacturing centers are working and which ones aren't, right? So there's only making the things it needs. You don't want to waste energy making stuff you don't need. So your body kind of has these self-regulating methods of shutting down pathways for things it doesn't need in a moment, right? So the one example we'll talk about in here is feedback inhibition, right? So kind of a common way your body does this is through feedback inhibition, right? So you remember, right? Enzymes are just working because the molecules they work with are bumping into them and they're doing their thing, right? And now we have these series of enzymes that are doing their thing. Something we get from the environment pops in here and here and here and pops out, and then we get the thing we need, right? Now, some of these pathways can be controlled if the end product, right, H in that first long chain, right, is also an allosteric inhibitor of the first enzyme, right? That's convenient because once you have a certain concentration of that end product, it's going to start bumping into enzyme 1. And if enzyme 1 has kind of an allosteric off switch, when you've made enough of the end product, it's going to start turning the pathway off and kind of regulate itself at some given capacity, right? So you get something that looks kind of like this, right? We have this long chain that we made. And so if we want H, We don't need like infinite H. At some point, we'll have enough of this little molecule that it'll start bopping back into this first enzyme up here, right? It'll be an allosteric inhibitor. It's going to stop this enzyme, which means we never get to the next step and the rest of the pathway never happens, right? So C's enzyme might have an allosteric site for H, right? Something like this, right? This is that same process. Initial substrate, reaction, 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 final substrate. But enzyme 1 has an allosteric site for the end product. And so once this plugs into the allosteric site, this is no longer, right, the right shape. First reaction never happens. Pathway shut down. Right. And we'll see when I think it's in photosynthesis. I think it's in both of them. We'll see examples of this kind of feedback inhibition, right? Where when you have enough glucose or whatever, the pathway kind of shuts itself off. All right, which is where we leave off chapter 6. Cool. So, big broad strokes for chapter 6. Chemical bonds are a primary location for energy storage, right? Chemical bonds hold energy. Breaking those bonds releases energy. That is kind of what your body's doing all the time, making and breaking chemical bonds, right? Our cells have developed pathways for making specifically the molecules you need, and also for breaking down the molecules you need broken down. And all of these reactions and all of these pathways are facilitated by enzymes, right? And so again, we'll spend the next 2 chapters talking about kind of our prime examples of these series of pathways, right? Respiration and photosynthesis, all right? And so that is where I'm going to leave it today. We'll kind of do a brief 6 review when we come in on Friday, but I'll let you go a few minutes early. We'll just kind of leave it there for today. So